Hello, welcome to today's problem session. Today we're going to be talking about the types of RNA, RNA processing, the genetic code, and translation. So let's get started. This chart is a chart that was discussed in your lecture, and I encourage you to go back and get details about each of these types of RNA. I'm not going to go into the details here, but some of these are ones that we'll talk about more than others. Certainly ribosomal RNA, messenger RNA, and transfer RNA are probably the three that I will mention the most. We will today talk quite a bit about small nuclear RNA because it's important in processing the primary transcript of messenger RNA to make it into the functional form. The lectures talk a bit about small nucleolar RNA, which is important in processing ribosomal RNAs. MicroRNA and small interfering RNA are both important in RNAi, which is RNA interference. I really won't mention Pee-wee RNA. We will talk about CRISPR RNA probably a little bit more later in the semester than we will now. And I won't really talk about long non-coding RNAs either. For each of the ones that I have listed here in the table, you should know what the abbreviation before RNA means. So for example, the R meaning ribosomal, and you should know a bit about what it does. For the ones we go into in more detail, of course, you'll need to know a little bit more about those as well. When we talked about transcription in eukaryotes, we mentioned that the primary transcript, the part that's formed from transcription, is not the same as the final messenger RNA that's sent out to the cytoplasm. And I briefly mentioned the three types of processing that occur. So let's look at these in a little bit more detail. The first type of processing is the addition of a 5' cap, which is a 7-methyl guanosine triphosphate, or GTP, that's added to the 5' end. And it's added, I'm going to say backwards, because it's a 5' to 5' linkage, which is not normally how you join nucleotides together. This is added early on in transcription, pretty much as soon as that 5' end is available. It's important for providing stability for the messenger RNA so that it doesn't get degraded. It's also important for the initiation of translation and for transporting the messenger RNA to the cytoplasm. At the 3' end, we get cleavage and the addition of a poly-A tail. So the 3' end is clipped off a little bit. And in fact, actually, I should have shown this diagram. This is the 5' to 5' linkage for the 5' GTP cap. But over here, we're looking at a site, and I'm not going to ask you to memorize anything about cleavage sites or consensus sequences, but there is a site after a consensus sequence where cleavage does occur. That cleavage occurs, and then the poly-A tail is added. And this could be hundreds of ribonucleotides, and they're all adenine residues, poly-A tail. This poly-A tail is very important for RNA stability as well. It also helps with the initiation of translation. And you say, wait a minute, translation starts at the 5' end and goes to the 3'. How does that 3' tail actually help with that? But it does, and so that's really important. The other thing is when RNA is degraded, that tail tends to get shorter and shorter. And once it gets short enough, the cap is removed, and then the whole thing really falls apart. So that RNA stability depends quite a bit on both the 5' cap and the poly-A tail. The last part of the processing of the primary transcript has to do with removing introns and joining exons. Introns are intervening sequences. Exons, I guess, are meant to be the opposite of an intron. So it's the part that's kept. And I realize that that is a bit confusing because you think of exon and exit, but try to remember intervening sequences, introns, are the parts that are going to be removed. These are non-coding regions, and they have to be removed prior to translation so that the proper messenger RNA sequence will be present for translation. The site where this occurs is called the spliceosome, SPL. And this is where the small nuclear RNAs come into play. Those small nuclear RNAs complex with proteins to form small ribonucleoproteins. And those are very important for holding the messenger RNA parts into position so that as the intron is removed, the exons are joined together. Now, this diagram is pretty complex, and you certainly don't need to memorize the details. I do want you to know that the small nuclear RNAs complex with proteins to form the small ribonucleoproteins, and that the cleavage occurs at the spliceosome. There are consensus sequences that are important here. The beginning of the intron always starts with G, and then from a DNA point, that would be a T, but since this is RNA, that's U. There's a branch point, and then the end of the intron ends with a G. And what happens on this is that the 5' site is spliced. It forms what we call a lariat, or think of that as a loop joining over here so that you see this loop formed. So the 5' part is cut first. It forms the lariat, and we think of this as a branch point, and those bases are not a true base pair. It's more of a joining. You can see the diagram here, which you do not need to memorize. Then the cut is made at the 3' end to remove that intron. Now this looks like, hey, wait a minute, how did you get this piece and that piece joined together? But trust me when I say the mechanism is such that those are held in close proximity, and that's where those small nuclear ribonucleoproteins come into play. The point being here, introns are removed, exons are joined together. At a spliceosome, there is a lariat structure formed by clipping at the 5' end, adhering at this branch point, and then the 3' end is clipped, and it allows the exons to join together. So after the messenger RNA is in the proper form for translation, it can go out into the cytoplasm and translation can occur. Before we look at the actual process of translation, let's look at the genetic code, because somehow the sequence in the messenger RNA, which is in RNA nucleotides, has to be translated into the sequence of amino acids to form a protein. The coding dictionary was established primarily by Nirenberg and Letter. There were some other experiments, including the Nirenberg and Matai experiment that occurred before, but the Nirenberg and Letter experiment finally figured all of the rest of the codons out. This is one of my favorite of the classic experiments, and let's see if we can make this diagram a little bit more visible. What they found was that if you had a ribosome and a trinucleotide, so 3 bases in a row, which they could make in any sequence they wanted, and the charge transfer RNA, that would be the RNA hooked to the amino acid, that you could get the structure of the trinucleotide, the ribosome, and the charge transfer RNA all bound together. And if you filtered the solution, this ribosome was big enough so that whatever was hooked with the ribosome would stay on the filter paper. Everything else would go through. So they did an experiment called the triplet binding assay. Got that written right here, the triplet binding assay. And for each of those trinucleotides, effectively each codon, they did 20 little tubes plus replications, and in each tube, a different amino acid was radioactively labeled. And the label they used for this was carbon 14, and that would radioactively label the amino acid. So for example, UUU, they would radioactively label in one tube alanine, in one tube nothing else, and then in a different tube, they would radioactively label leucine. In a different tube, they'd radioactively label phenylalanine, and so forth, till they had all 20 of those amino acids radioactively labeled in different tubes. Now, each of the tubes would have the charge transfer RNAs in them that didn't have radioactivity, except for, of course, that one that was radioactive in that one tube. They would allow the structure to bind. The ribosome would bind with the appropriate charge transfer RNA with the appropriate amino acid. If that happened to be, and then they would filter the solution, if that amino acid happened to be the radioactively labeled amino acid, then the filter paper would be radioactive. If not, the filter paper wouldn't be radioactive. So for each trinucleotide, although there were 20 tubes, each with a different radioactively labeled amino acid, only one would actually result in a radioactive filter paper. And that would be the one where that trinucleotide coded for that amino acid. Print on this is a little small for me to see. GUU. So it looks like GUU was our right here. That says GUU. And that was our trinucleotide in this particular experiment. And so the amino acid that would bind with that was valine, VAL. And so this is the transfer RNA hooked to valine. And if valine was a radioactive amino acid in that particular tube, the filter paper would be radioactive. So with all of the 20 different tubes that coded for GUU, the only tube where the filter paper was radioactive was valine. And that indicated that the codon GUU coded for valine. The researchers did this for all of the different codons, and they were able to come up with a coding dictionary where of the 64 possible codons, 61 coded for a specific amino acid. There were 3 codons, UAA, UAG, and UGA, that did not code for an amino acid, and those are considered to be stop codons. So a couple of things about this chart. This is the coding dictionary that you will be given on exams. Typically, it's the first item on an exam if I feel you will need it on the exam. It's always set up the same. So the first position is the one on the left, UCAG. Second position across the top, and then within each of these boxes is the third position. So UUU codes for the amino acid phenylalanine, and the 3-letter abbreviation for phenylalanine is PHE. The 1-letter abbreviation is F. If you are asked for the name of an amino acid, it's okay to give the 3-letter abbreviation, and that way spelling won't accidentally catch you on Moodle. If you are asked for the 1-letter abbreviation, you must give the 1-letter abbreviation because that's how Moodle has programmed the answer. So let's take a look at working some problems with this coding dictionary. The one thing that's kind of interesting, if you just look at the coding dictionary for a second, one thing that we say is that the code is degenerate. That means that multiple codons can code for the same amino acid. And there's a pattern to the degeneracy. Notice that there are several of these where the first 2 letters are the same and the last letter differs, and they all code for the same thing. So AC anything codes for threonine. GU anything codes for valine. This pattern to the degeneracy is something that was noticed early on, and it's thought that possibly that third position is just not as critical in the binding, and we say that this is wobble in the anticodon. Now, there are some specific wobble rules that you'll go into in more detail if you take a molecular genetics class. We're only going to go into the one involving inosine. So inosine is an unusual base that's found in the anticodon of transfer RNA. And when it's in the anticodon, it's always in the first anticodon position. Now remember, DNA is written 5' to 3', RNAs are written 5' to 3'. So the first anticodon position is the one on the most 5' end. And I'm going to use a capital I to represent inosine. Inosine's kind of interesting because it can bind with every base except guanine. So it can bind with adenine, cytosine, cytosine, and uracil. So let's see if we can use inosine to explain some, it won't explain all, of the degeneracy. So for example, say I had the anticodon IGC. We might wonder what amino acid would be on that particular transfer RNA that contained this anticodon. So the easiest way for me to think about doing this is take the anticodon and rewrite the anticodon. So it's 3' to 5'. And I do that because then I can think of codons being 5' to 3'. And that's easier for me because on our coding dictionary, our codons are written 5' to 3'. So let's see. IGC, next to the 3' end is C, then G, then I. So that means the codons would have to be anti-parallel, 5' here, across from 3', and complementary. Across from a G, we'd have across from a C, we'd have a G, across from a G, we'd have a C. And then remember, I combine with A, C, or U. A, C, U. So GCA, GCC, and GCU would all be codons on the messenger RNA that would bind with a transfer RNA that had this anticodon. Well, let's take a look at our coding dictionary. Let's see, G, C, U, C, and A. Look at that. They all code for alanine. Inosine in the anticodon does not explain all of the degeneracy of the code. However, it does explain quite a bit. So it would explain the part where UC, UC, and A all code for serine. It would not explain UCG coding for serine. It would explain the part where CC, UC, and A all code for proline, but not why CCG would code for proline. However, there are some other wobble rules that will help to explain the other parts of the degeneracy of the code. Let's see if we can translate some things. I'm going to keep my coding dictionary kind of handy though. We've got a messenger, we have a DNA strand because it has T, not uracil, so we know this is a DNA strand. It says if this is the coding strand, what would the sequence of the messenger RNA be? Now, remember the template strand is the one that's anti-parallel and complementary to the messenger RNA. That means the coding strand looks exactly like the messenger RNA, except where there's a T, there's a U. So we've got a few places that have T's. All of those would be U's. So our messenger RNA looks just like the coding strand, except where there's a T, there's a U. And that's really a good thing to remember because oftentimes you see the coding strand of the DNA, and if you can mentally just flip those T's into U's, then you can read it as a messenger RNA and use the coding dictionary. So let's see if I can write that out. CCU AGA, UGCG, CAAC, UCUG, CCU, AGC. All right. Didn't quite fit in the box there. And now let's see what we would do to translate this. To translate, always start reading the messenger RNA from the 5' end. Read along until you find a start codon. Now, I didn't make this clear when we were going over the coding dictionary. I mentioned our 3 stop codons, but the start codon is always AUG. You can remember that the start codon is the only codon for methionine, and that means when we translate, the first thing that's put in is methionine. Or I like to remember school starts in August, and that's the start codon. So I will go along here, AUG, there's a start codon. And I will break it into 3-letter codons, CGC, AAC, UCU, GUU, until I hit a stop. UGA is one of the 3 stop codons. So this means my protein will have 1, 2, 3, 4, 5 amino acids in it. Proteins are made from the amino end to the carboxyl. So you can think of this as the N terminus and the C terminus. You can think of it as amino and carboxyl. You can think of it as NH2 to COOH, and all of those are abbreviations that I've probably used at some point in your slides. The first one, AUG, go up to our coding dictionary. We know that codes for methionine. CGC, CGC codes for arginine. AAC, AAC codes for asparagine. UCU, UCU codes for serine. And GCC, GCC codes for alanine. So we've got our protein: methionine, arginine, asparagine, serine, alanine. If you had needed to do the 1-letter abbreviations, that would be M, R, N for asparagine, S for serine, and A for alanine. Going from the N to the C terminus. Okay. Let's take a look at this next problem. Here we've got a double-stranded DNA molecule that's missing a few bases. So we need to fill in what these bases would be. We've got the ends labeled 5' and 3' on both sides. Even if you only had one side labeled, remember the other one's easy to label because you know if it's a 5' end there, it's a 3'. You know if it's a 5' end here, this has to be a 3', which means that would have to be a 5'. So as long as one end is labeled, you should be good to go. Now this is DNA, and we want to fill in the bases for DNA. So A, T, G, C, G, A, C, A. There we go. So now we've got our DNA strand made. Now let's say the entire transcript is transcribed from left to right. And we'll pretend all the appropriate control signals, the signed Dalgarno and so forth, are all there. So we want to label the messenger RNA, and we also want to identify which strand is the template and which is the coding strand. So if transcription goes from left to right, then our template strand has to be read 3' to 5' in the direction of transcription. So the lower one is the template strand and the top one is the coding strand. And then we know, just like we did before, the coding strand, by changing the T's to U's, is the correct sequence for our messenger RNA. So I'm going to start a little bit further over here so I can write it all out. CGGAUG. CGACAG. CGACAG. UUCAGA. CAUUAA. UACU. So there's our messenger RNA. Now we need to translate that messenger RNA to get the protein sequence. Again, we start at the 5' end, go till we find a start codon, AUG, break it up in 3-letter codons, until we find a stop. And it's going to be convenient if you can remember what these stop codons are, UAA, UAG, and UGA. However, remember, you're always given the coding dictionary, so you can look them up. Or if you know you're going to be doing a problem like this, you can always start off by saying, oh, I'm going to write on my scratch paper, here are my stop codons. And then when you're breaking this up, you can identify it pretty easily. We now need to translate this, going from amino to carboxyl. And I'm just going to write the first letter, or the 1-letter abbreviation for each of these, just so I'll have a little bit more space. So AUG codes for methionine. CGA codes for arginine. It's R. CAG codes for glutamine. Q. UUC codes for phenylalanine. F. UUC. AGA codes for arginine. R. G. And CAU codes for histidine. H. Oops, there we go. Now you can see. So this is using the 1-letter abbreviations: methionine, arginine, glutamine, phenylalanine, arginine, and histidine. Be careful when you're doing your homework. If Moodle asks for the name, make sure you give the 3-letter abbreviations. If it asks for the 1-letter abbreviations, make sure you give the 1-letter abbreviations. All right, let's try another one. So this one says, given the sequence of the template strand of DNA, what's the sequence of the protein? Now this one's a little bit different. It says, assume this is in the middle of the gene, so you won't necessarily see start and stop codons, and make sure you label the ends of your protein. So this is a template strand. Now remember, when we're making RNA from the template strand of DNA, it's easiest to read the template strand if we write the template strand 3' to 5'. Now it's written 5' to 3' here because that's our convention. If you're writing only one strand of DNA, you write it 5' to 3'. So if I wanted to flip this around and make this a little bit easier, there are 2 ways I could do it. I could transcribe it first, or I could flip this. So my template reads 3' to 5'. And that just goes GAT. And these are broken up in the codons. I'm going to call this template flipped. Then AGG, CAG, CCT, and GTA. Now, when I write my messenger RNA, my messenger RNA will read 5' to 3' in the direction that I'm used to going, which for me is left to right. So anti-parallel and complementary. Across from G, I would have C. Across from A, remember this is RNA, so it's U. Across from T is A. Then we have UCC, GUC, GGA, and CAU. And now I can use my coding dictionary to translate that. So remember, we're going to go from the M terminus to the C terminus. CUA is leucine. UCC, there we are, serine. GUC, valine. GGA, glycine. And CAU, histidine. So our protein should read from the N terminus to the C terminus, leucine, serine, valine, glycine, histidine. And if I wanted that in one-letter abbreviations, leucine, L, serine, S, valine, V, glycine, G, and histidine, H, and then the C terminus. So this N is for the N terminus, that C is for the C terminus. And then these are our amino acids. Okay. I think I have one more question here. Let's see. Here we go. This one involves inosine. A transfer RNA anticodon has the sequence inosine, adenine, cytosine. What is the amino acid that is carrying? So we need to figure out what codon this would bind to. So if I think about anticodon, IAC. So I'm going to write the anticodon from 3' to 5' so that when I write my mRNA codon, it will be 5' to 3'. So C is near the 3' end, A, I. The messenger RNA codon should be anti-parallel. That means the 5' ends here, and complementary. So across from C, I have G. Across from A, I have U. And then this would be either adenine, guanine, or uracil. Oops, not adenine, cytosine. Cytosine, not guanine, because inosine does not bind to guanine. It binds to adenine, cytosine, or uracil. So my 3 codons are GUA, GUC, and GUU. And I'll look on my coding dictionary, and GUU, C, and A all code for valine. So the name of the amino acid that this transfer RNA is carrying is valine. And then these are the 3 codons. And if we want to label the 5' and 3' of each of these, here we go. Now they're labeled. I hope this has been helpful. Please contact me if you have any questions, and thank you for your attention. Have a wonderful afternoon.